A very good afternoon to all. I'm Shripriya Singh and I'll be presenting targeting melanocortin type 2 receptor MC2R with antibodies and peptides for the treatment of congenital adrenal hyperplasia. To briefly present the background, congenital adrenal hyperplasia CAH is a common disorder of steroid production. It is caused mainly by the mutated CYP21A2 gene, which mainly causes the 21 hydroxylase deficiency. So in this, what happens is that patients require a lifelong therapy with mineralocorticoid and glucocorticoid supplementation. However, due to the location of the enzyme block in the pathway and the increased production of the adrenocorticotropic hormone, which is the ACTH, it stimulates the MC2R receptor, and therefore excessive androgen production remains a major challenging side effect. So if we want to negate or somehow nullify the side effect, we need to block or inactivate the MC2R receptor in patients with CAH. And if we are possible to do this, it would be a very specific approach for treating the androgen excess in CAH without affecting gonadal steroidogenesis. So for the approach and methods, what was the approach? First of all, we need to know the three-dimensional structure of the receptor as well as ACTH. And once that is in hand, we model the interaction of ACTH with the MC2R and design specific peptides and antibodies to understand the ACTH binding and activation of the MC2R. So using this new model, we've been able to design peptide sequences for producing antibodies specific for MC2R. I'd also like to mention that this took a number of rounds. So we had like 3 to 4 rounds, and at every step we tried to improvise the design of the peptides. And then now we are at the 4th step, and so we have like good peptides in hand, and now we can proceed for further studies. So finally, we docked the ACTH into the MC2R structure, and using molecular dynamic simulations, we improvised the structure and the interaction and analyzed the binding of ACTH to the MC2R. Next, we have, these are some of the structures that we have, like we use softwares like Autodoc Vina and Yasara. So we have a receptor here, which is the MC2R. We have ACTH, which is again the structure here. And then we have an interaction complex, which is MC2R ACTH complex. Now, in order to design the peptides, we need the smallest peptides. So it would be like the ACTH sequence is well known. It is a 39 amino acid sequence, but we know that the first 24 amino acids are crucial for the functionality, and the rest 15 did not have much effect. So now we want to choose the peptides which are shortest peptides, like let's say 4, 6 or 8 amino acid sequence, which will act like dummy ACTH molecules. They will go and specifically bind with the receptor, and they will not have the functions of ACTH. So it will be like an inhibitor. Now to do that, we had to study what is the interaction, what are the best peptides. So we perform a sequence tolerance plot. In this, what happens that we study each and every interaction within the residues and between ACTH and MC2R. So these ones in red, they are the naturally occurring amino acids. And because we can see that they're ranking at the top, they are the best. So while we're designing our peptides, we must retain them, and we can play around with the other sequences. So this plot can be used to strengthen the efficiency and the activation. That is, we want a peptide which binds well, but does not signal like ACTH. So we can weaken the signaling part, but we can strengthen the interaction. So on this basis, we chose a 4, 6, and 8 linear peptides. These are the sequences. But now we wanted to improve the design of the linear peptides, so we wanted to circularize them. Circular peptides are much better in terms of their binding and in terms of their stability. So we introduce the cysteine residues, which will add the sulfhydryl groups, and these sulfhydryl bonds will now circularize the peptides, and these are the peptides that we want to screen. The second specific approach would be to target the ACTH MC2R with specific antibodies. But now these antibodies should be so specific that they do not target the other receptors. They should be very specific for only the MC2R. But because the melanocortin receptors have sequence homology, we had to sequence all the amino acids and check that there are not overlaps. So then finally we identified 2 unique peptide sequences in the MC2R that are absent in the other receptors. So here we have the sequence of the peptides, the amino acids, yes. And then finally, the structure and also the uniqueness of the sequence is that it is a part of the ACTH binding site and therefore will serve as an excellent target for developing anti-MC2R antibodies. Finally, when we've done the dry lab part, finally we perform the in silico part. Now the question comes, where are we going to perform the experiments? We need a wet laboratory scenario. We need a cellular system. So for that, a number of experiments were carried out with 2 to 3 cell lines. And finally, the mouse OS3 cell lines, which is the mouse adrenal cell lines, showed the best effects. So we transiently transfect them with the receptors, we stimulate them with ACTH, and then we can study the effect of the peptide or the antibody via dual luciferase assay. So this simply demonstrates that the testing of the signal response is established in a mammalian cell-based system, and it is ready to be used anytime we want to screen a peptide or an antibody. Next, we also wanted to check the effect of the designed peptides on the other receptors. So here we have chosen one sample peptide which gave us best results, which is the GK10. And we saw the effect on the different melanocortin receptors, MC1R, 3R, and 4R. And it was seen that against the decided peptides, some activation of MC1R was observed, but there was minimal cross-reaction with the MC3R and MC4R. So it means that we are narrowing down to a peptide which is going to be more specific for the MC2R and not the other receptors. Finally, this data basically shows that the experiments were carried out in the different cell lines, like we had HEC cells, we had COS cells, and we also had the mouse OS3 cells. And these cell lines were tested for transfection and activation, and OS3 cells showed the best results with minimum constitutive activity of the receptors. So therefore, OS3 cells have been chosen for later studies, and all related MC receptors are established in a mammalian system, and we can simply proceed for the experiments. Finally, we also wanted to check whether the peptide is acting like ACTH or it is acting just like an inhibitor, because initially I said that we don't want a peptide which will have similar signaling properties like ACTH. So we compare, and these are the different peptides which have been compared, and we can see that here H38 is one of the best peptides for us because it has a result which is very similar to control. It is not activating the MC2R receptor. So here we can use this as a potential inhibitor in our future studies. So with this, we have a number of assays in hand, and now we're trying to improvise on the designs and check these peptides and then finally move to the antibodies. And then to conclude, the novelty and the significance of our project lies in the fact that so far there is no treatment available to block or inactivate the MC2R receptor. And therefore, we can solve the problem of the side effects of the androgen excess, which is possible in CAH. And if we are able to do that, we can easily change the profile of a CAH patient to the profile of an FGD patient, which is a familial glucocorticoid deficient patient. They also are treated similarly because they're also given a mineralocorticoid and glucocorticoid supplementation. However, they do not have the side effects like early puberty, virilization, and bone maturation. So somehow, if we have a peptide in hand, and the smaller the peptide, it will serve as a novel drug candidate in future. Or if we can have an antibody, we can further move on to the humanization and finally commercialize it. So if we have this kind of a drug or an antibody in hand, we will be able to contribute significantly in the treatment and the medical fraternity also. So we will have like a possible solution for treating these side effects in CAH. And inhibitors of MC2R signaling and specific antibodies will advance the field of possible treatments of androgen excess in CAH. With that, I conclude, and my sincere thanks to Dr. Pandey, Professor Fluke, and Dr. Shahina Parveen, all my funding agencies, and especially to ESCO Scholarship for funding my stay here in Switzerland. So with that, I say thank you.
Thank you very much, Dr. Singh, for your presentation. So I wondered, listening to your talk, you have now identified, for example, this H38 peptide as a potential candidate. Did you already test whether it has a similar effect as a competitive inhibitor on MCR4 or MCR3? because if you block the action of these receptors, you may have other problems that emerge, such as obesity or weight gain, given that mutations in these receptors are associated with obesity phenotype. Can you develop your thoughts on this?
Absolutely. We have actually narrowed down on this peptide and we have checked it for the other receptors also. I haven't shown the data here because we are focusing more on the MC2R, and I also had like a little time constraint for 10 minutes, but we've already checked and seen that it is not having a non-specific targeting action. So that is now we're working on it and we're trying to improve the peptide designs and check for more peptides.
So in other words, the MCR3 and MCR4 signaling is absolutely unimpaired by your peptide. Is this
Yes,
correct?
the effect is not significant. Like we can negate it and then we can take this peptide and try to improve the design of it further so that we do get results. Like a little bit is there. I would not say it is absolutely unaffected, but the results were not significant. So we can say that it is not affecting it too much.
What do you mean by non-significant? Now, if you have a decrease in signaling, even if it is minor, it could certainly have a metabolic effect. How did you define what is significant and what is not significant?
It is only a little mathematical when I say you're absolutely right, because for signaling, even minor changes can be very important. And that is why we have just chosen this peptide. And now we're trying to modify the residues further and check
Okay.
again so that we get no effect. So it is just like shortening. It's like funneling down to a peptide. Yes.
Okay. And just for my own education, is there any constitutive activity in any of the MCRs? And do you silence, if there is constitutive activity or basal signaling, do you silence some of this with your peptide or not?
No, that's a great suggestion that you're giving, because no, we've not tried that yet. Like exactly the constitutive, we've not checked it completely. Yes.
Okay, because some of the GPCRs do have constitutive basal activity, and I think that's also, I don't know the details for the MCRs, but I think it would be relevant also to at least check on this if it is present.
Absolutely. That's a great suggestion. I will definitely do that in my future experiments also. Thank you.
Okay, so we thank you a lot for your presentation. We will close here. We will bring the presentation in shape for the website, and I wish you all the best for the continuation of your work.
Thank you so much. Thank you. It was a
Okay,
pleasure.
bye-bye,
Thank
Dr.
you.
Singh. Thank you.
Thank you. Thank you.